Every day, there are invaders trying to kill your body, or at least use your body for their own personal gain. And it usually makes you feel under the weather or sick. So how do we stop this from happening? Well, we have a wonderful immune system that constantly tries to stop this. What's our first line of defense? Well, our skin, our tears, our sweat. Yes, all of those things help keep invaders from getting into your body. So what happens when we breach those barriers? When we eat food covered in invaders, or trip, or cut ourselves, what happens then? Well, if you remember back to the integumentary system, our body tries to heal itself. But during that time, the walls are being rebuilt, we have to send soldiers to maintain control. These cells are the brave lymphocytes, or more commonly, white blood cells. They are constantly rolling through our bloodstream, getting signals by selectins and adherins to move into the site of invasion. Well, how does this get initiated, you may ask? Well, when our body gets damaged, signals called DAMPs are released. And when invading pathogens enter our body, they have certain PAMPs. And when resident immune cells that are always in the area sense these, they light the beacon for help. What is this beacon? Well, they're chemokines that the immune cells release, which signal the endothelial cells to put out adherins on their cell surface, as well as loosen the tight junctions between themselves so that our brave heroes can fit through. Once the white blood cells have made it through this barrier to their destination, they've entered a battle zone. We'll be fighting off the terrible invaders one at a time. So how do we destroy these invaders one by one? Well, our brave heroes have a couple of ways of destroying the enemy. Our natural killer cells can kill any cells that have abnormal plasma membrane proteins. They do this by secreting granules called porphyrin that can create holes in the membrane and the cell will burst. This is a process called cytolysis. Another option is that macrophages in the blood can come through and eat the smaller pathogens, a process called phagocytosis. Phagocytosis takes place in 5 easy steps. Chemotaxis, where the phagocyte moves towards the site of damage. Adherence, where the phagocyte binds to the invading pathogen. Ingestion, where the phagocyte extends pseudopodia and engulfs the microbes into a sac called the phagosome. 4. Digestion. The phagosome merges with the lysozyme, and this breaks down the microbial cell walls. 5. The chemical slaughter quickly kills many microbes, and they can be broken down further by residual bodies. So what happens if the invaders are too many and our brave heroes, the macrophages and the natural killer cells, get overrun? The next step is to call in our special forces, or our adaptive immune system. Our innate cells can migrate via afferent lymph pathways to the secondary lymph nodes, carrying an invader or an antigen. By the time they get to that secondary lymphoid organ, or our lymph nodes, have become an antigen-presenting cell. They find the nearest general, or T cell, and present antigen and co-stimulate the T cell, which can recognize exactly which antigen we are dealing with. The cytotoxic T cells will be activated if the pathogen is intracellular. Then they will migrate via efferent lymph nodes to the battle zone, or the site of inflammation, and destroy all of our infected cells. If the microbe is extracellular, Then the helper T cells will become activated and activate B cells to produce antibodies. The antibodies being produced by the B cells come in 5 flavors. We have IgG, which is the most abundant of the antibodies and is found in the blood, lymph, and intestines. It helps protect against bacteria and viruses. IgA is found mainly in sweat, tears, saliva, mucus, breast milk, and GI secretions. It protects the mucous membranes against bacteria and viruses. IgM is the first antibody to be secreted by the plasma cells, or mature B cells, and causes agglutination and lysis of microbes. IgM can also be found on the surface of B cells, where they serve as antigen receptors. IgD is found on the surface of B cells, and it is involved in activation of B cells by T cells, or antigen-presenting cells. IgE occurs as a monomer and is involved in allergic and hypersensitivity reactions. It also provides protection against parasitic worms like helminthes. Our 5 flavors of antibodies also serve 5 main functions. They can act as neutralizing agents by preventing bacterial toxins and viruses from attaching to our cell surfaces, which prevents them from proliferating. They can immobilize bacteria by binding onto their flagella and making them incapable of moving and spreading into nearby tissues. And precipitate antigen, where the antibodies clump together the pathogens so that phagocytic cells are served their dinner on a silver platter. They activate complement, a cascade that pokes little tiny holes in the invaders until their intracellular fluid looks just like their extracellular fluid, which is very incompatible for life for those cells. They can enhance phagocytosis, a process called opsonization. They basically bind to invaders and put up a big red flag for the phagocytic cells to come by and gobble them up. So these are the main ways that our immune system is fighting the invaders that are constantly trying to get into our body. This is why we walk around every day, not always sneezing and sniffling, feeling good, studying anatomy, learning things.